So good afternoon, everybody, and welcome to this new online technology meeting from EPIC. My name is Antonio Castello. I'm the technology manager at EPIC, in charge of all the laser and biomedical topics. And it's a pleasure for me to be here to talk about what is new in photonics development for diagnosis and treatment in ophthalmology. So we will talk about different developments and solutions, photonics related, that can be applied to the different segments of the eye, and also related with intraocular lenses. That is one of the hot topics in ophthalmology. So first, a few words about what is EPIC. For those of you who doesn't know, EPIC is an association of more than 800 companies and research centers, all of them related with photonics. The name EPIC, European Photonics Industry Consortium, summarizes our goal, that is improve the business of the photonics industry in Europe. And as a member of the EPIC staff, our also goal is to help all the members with the different requests they may have. So we support our members in having access to different macro reports to understand what's happening in the new markets and applications that they are interested in reaching or understanding more what's happening there. Also, we can help with the issues related with technology. Can be a technological need in terms of a component that they are missing, a new solution, perhaps understanding alternatives to the components they are using nowadays. So we support our members also to find new collaborators and new partners in the technological issues. We also help with other more business-related topics. So we have a Job in Photonics portal to support with the human resources activities. We promote the job openings. That is a topic that several members mentioned, that is sometimes difficult to find people with the right skills and the right experience. So we try to promote these open positions, and also in different exhibitions and trade fairs to make people aware of all the opportunities within our members. We also have a list of investors and a list of companies interested in invest, for those of you that are interested in perhaps going in this direction, meeting venture capital companies, talking with these venture capitals and investors, and understand what are the requirements to work with you, and also the other way around, small companies that are interested in being acquired or be part of a bigger group. We can put them in contact with members that are interested in expanding the group or their portfolio of solutions by acquiring a company or a technology. And the last pillar is the networking, and that's one of the topics that why are we here today, is to meet people in the field of ophthalmology related with photonics, understand what are the companies doing, and also interact with all of the speakers. So this is why we will have time after each presentation for Q&A. So I encourage you to switch on your camera, raise your hand, and also make your question directly to the speakers. You can also use the chat in case you're not allowed to use the camera, and I can also help by sending your question to the speaker at the point. As part of this networking activities, we are actively working in different events during the year. We are already working in the 2026 agenda, but for the moment, perhaps it's interesting for you to focus in the 2025. Of course, part of our agenda, I would say most part of our agenda in the next weeks is focused on the events that will have Laser World of Photonics. That is one of the main shows that we have. In this case, every 2 years, all the EPIC team will be there, and we will have a lot of different meetings, coffee, breakfast, sorry, with the CEO of the different companies. We will have marketing coffees. We will have events related with sustainability, a small meeting also related with human resources. And related with technology, we will be part of the integrated photonics application stage. So my colleague Ivan will be moderating a few sessions of the use of integrated photonics and applications in medical, bio, and also telecom, datacom. So you can check all the presentations there if you're interested in this field. We will have another technology meeting focusing innovative optics, and another one in laser micromachining applications. That we will have very interesting presentations about the different technologies, and we will have finally a workshop at AMS booth about the solutions for this topic. And back then in September, we will have a new delegation trip to Taiwan, and we will start also the second part of the year, let's say, or after summer part of the year, with a technology meeting on industrial laser processes in Italy. And we combine this with online meetings. Of course, as we have this one, in July, we covered, let's say, the lack of travel because some of you will be on holiday. So, but we will continue activities for the ones that are working, interested in knowing more about photonics for the smart farming. We will have also the second discussion about the impact of US tariffs. That is a topic that is, let's say, worrying several of our members. And the first one was an interesting starting point to discuss the experiences of the different members, how to put solutions in potential problems one member may have. So I recommend you to take part if you are involved in business with US, that I think most of the members are, and understand from the experience of the others what's happening there, what can happen, and perhaps potential solutions for the issues. And then in September, we will continue with the technology meetings in different topics like quantum and mid-IR developments and metrology systems. Most of the companies here will be or are related with biotechnologies, bioapplications. So I want to highlight two of the meetings that we will have in October and December. The first one is related with photonics for bio and life science applications at CSCM Neuchâtel. In this case, we will talk about different spectroscopy, imaging solutions, analytical, bioprinting, biosensing that can be used in medical, bio, food industries, pharmaceutical industries. The idea is to put in common with end users, integrators, and the components manufacturers, learn about the new developments and understand also the challenges and needs for the future. And in December, we will have our traditional meeting on cancer pathology and therapy. In this case, with Gustave Roussy Cancer Center in Paris. And the goal is to bring a good number of medical doctors to talk about their experience with photonics technologies, their actual work, and a good point of interaction with photonics industry to find potential collaborations, potential hospitals and partners where to start the clinical trials. I think it's a good combination of challenges from the doctors and new developments and partnerships for the future that it have been quite successful up to now. So we are very happy to visit Paris this year with this topic. And regarding today, this is the let's say summary of the companies that are registered. So it's good to have a good number of companies from the different topics. So we'll have companies that are more involved as end users and integrators in ophthalmology, also manufacturers of specific solutions and systems for this field. Of course, components and modules, optical components, coatings, lasers that are needed. Metrology and testing solutions is also quite important in this field. In the case of intraocular lenses, for example, it's important to have a good metrology system, good characterization system. And then we combine with material service providers, also investors and consultants. So a good distribution of companies interested in this week that is a little bit complicated because it's just before LASIK World Photonics in Munich. Yeah, we can start with the agenda. We will start with the first presentation. This meeting is sponsored by our member Tecnottica, so I want to thank you, Tecnottica, for also supporting our activities. And I will talk with Tecnottica at the end, so they can have a presentation. So I will ask you to stay at the end, so they can show us in one slide their capabilities. This is the agenda for today. So we will start with a presentation from Maculaser. We will continue with Imaginize, then Ansys, Bausch Lomb, Lumos Laser, and as I mentioned, we will finish with the slide by Tecnottica to show their capabilities. So this is everything from my side. So I want to introduce you Gianni Tironi, that is the CEO of Maculaser, and he's going to present the solution for restoring retinal health with photonics. So the floor is yours, Gianni. You can start sharing the screen and start with your presentation as well.
Yeah, thank you. Just a second.
Yeah.
All right. Thank you for inviting us to present here today. My name is Gianni Tirronen. I'm the CEO and co-founder of Maculaser. Maculaser is a clinical stage deep tech medical technology company originally from the leading vision research laboratory in Finland. So we are developing device, a new method to treat retinal diseases. And the biggest unmet need in the ophthalmology is called age-related macular degeneration, a retinal disease that have only 5 to 10% of the cases have some treatment available. It's divided traditionally to two different classes. It's dry AMD and wet AMD. Wet AMD is 5 to 10% of all cases, and that's currently mostly treated with anti-VEGF intraocular injections. There are many potential causes for this disease to happen, but the biggest one, oxidative stress, drives progressive retinal neurodegeneration. It's a leading cause of blindness, over 200 million affected globally, and a staggering 300 billion annual direct healthcare cost. That's approximately 1/4 of the Alzheimer's disease healthcare burden. In this disease, typically you lose the central field of vision gradually. And one more thing, retina is located in the back of your eye. When talking about eye lasers, it's typically something happening in the front. I mentioned the various pharmaceutical solutions, but the main problem is mostly unsolved. Even the anti-VEGF drug market is over 12 billion euros annually. Non-damaging lasers, unlike photocoagulation lasers, they've been around 10 to 15 years so already. It's been shown in various over 100 clinical trials already, the effects that heating activates the therapeutic targets essential for AMD management. And these targets are heat-shock protein A production, autophagy activation, and antioxidant stimulation. So in this treatment modality, you heat up the retinal tissue mildly without causing damage. And it's very important that the actual temperature in the retina is within the certain limits. The problem so far is that consistent therapeutic retinal heating cannot be achieved without thermal dosimetry, meaning that it's almost impossible to heat consistently with its target temperature. And that's because people are different. There's different pigmentation, blood circulation. Also, elder people have ocular opacities. So it can cause over 4-fold variation in retinal temperature when you use the same laser power. But we estimate that with the current treatments, only approximately 20% of all patients will get some benefits. So now we are getting to our solution. We have developed technology to measure the target temperature in the retinal tissue real time. How do we do it? We actually use 4 different light sources. In addition to laser, most important one being the flashes of light. It's called electroretinography system, which is typically used for diagnostics. We don't use that for diagnostics, but as part of the treatment. So there are flashes of light. These flashes generate electrical response in photoreceptors in the back of your retina. We record that signal or electrical response with our sensor, then use our algorithms to transform the sensor data into temperature info. And when we know the temperature in real time, we can adjust the laser power accordingly, personalized treatment, and reach the target temperature in every individual, but also within the same retina in different treatment locations. Electroretinography is well-known diagnostic system, but we don't do anything separately. Everything is embedded in the same device and seamlessly to the treatment flow. So the actual product, it's a non-damaging retinal laser with thermal dosimetry. It's pretty traditional setting here, used in a retina specialist office environment, class 2 medical device. Only difference to traditional photocoagulation lasers in a patient setting is that this is a non-contact system, which adds some, it's more complicated technology-wise, but very useful for the patients. So yeah, a couple of features here. I said we are clinical phase startup, so unfortunately we cannot share everything in our technology side, but some of the target features: non-contact laser solution. It's patient-friendly, and also for doctors doesn't need to touch the patient anymore. So that all the treatment beams, 4 different light sources, are directed to the fundus via fixed non-contact lens. And second feature, as mentioned, we use electroretinography signal to reach the target temperature, which is the main feature, and which enables the treatment efficacy and safety. But the same signal also enhances safety. We can go much higher temperatures, treatment temperatures, which is more efficient with online safety mechanisms. So we actively monitor the temperature, but also the signal, which tells us the kind of when we are close into the lesion temperatures. And part of the system is also the automatic pupil tracking, extremely fast. So it ensures the correct device alignment, and also if there are some excessive eye movements, halts the treatment. So as a summary, the key thing is, of course, to try to find the effective treatment for retinal diseases. But also, when we have developed a solution, there are several different other innovations that we are utilizing here, and kind of stepping up with some sort of traditional fields to show the patient safety, but also the comfort. So I was asked to prepare some questions, what we can offer to you, or they, and what they can offer to us. The whole system is pretty complicated, utilizes lots of different photonics related technologies and applications, optics, eye tracking, then the consumable sensor, which is an eye electrode, manufacturing lines, biocompatibility. All these would be welcomed if there would be some cooperation, and also the grant funding joint applications we are interested. Yeah, so we might be able to offer some joint projects and perhaps even commercial cooperation. So this is my presentation. Thank you.
Thank you very much, Yanni. So we have now time for questions. So as I mentioned, it would be great if you can raise your hand and make your question directly to Yanni. We can start with a question we already have from Tecnottica. I don't know if you can do the question directly. In parallel, they are asking which type of lasers are you using nowadays in your solution?
Yeah, we are not developing laser technology ourselves. It's everything what happens after the laser, but the laser is 810 nanometer infrared laser.
What's the level of power? Because you mentioned that you control the power of the laser with this sensor, but what's the level of power normally you are using?
Yeah, we define it as milliwatts per millimeter, which is the radius of the treatment spot. So typically it's somewhere between 100 and 200 milliwatts per millimeter, and the treatment spot can be anything between 2 to 5 millimeters.
Okay.
So it's a quite high powered
Yep.
laser.
Patrick.
Yeah, Yanni, thank you so much for your talk. Really interesting technology. I had a question a little bit on at which stage of AMD are you planning to, or is it meant to be applied? Is it at early stage? Is it at late stage, or in the transition towards it? And a
Yeah.
little bit more on the clinical outcomes that you see as a result of the treatment.
Yeah, I mentioned these 100 other clinical trials that have been done with the non-damaging lasers or sub-threshold lasers. They show efficacy in every stage of AMD, but our clinical trial plans, we actually have a kind of 2 major trials upcoming starting next year. First one is actually for wet AMD, so it's combination therapy. There will be one anti-VEGF drug injection first, and then we will replace the next injections with our laser. Because there have been lots of studies that, I forgot to mention that even the anti-VEGF works pretty well. Almost 50% of the patients stop responding after 2 years to that drug. And there's evidence that actually this kind of laser therapy, you could kind of continue giving these injections much longer and keep these people responding to the drug. So wet AMD is a late stage AMD. Second trial, 140 patients, that's for dry AMD. It's intermediate AMD, but we plan to include some of the early GA patients, which are geographic atrophy patients. One of the commercial reasons there is actually the Apellis FDA-approved GA drug that was refused in Europe last year. We want to compete directly with them because they didn't show any efficacy in vision improvement, but still it's 600 million business in US already. It slows down the geographic atrophy growth mildly, approximately 20% a year. But we believe that there are good reasons to believe that our efficacy would be better than theirs. But the short question is, our targets are the intermediate AMD and then also the wet AMD. There are also other retinal diseases like diabetic macular edema. Actually, the kind of evidence there is strongest one of all, but also the competition there is very big. So that's why the AMD.
Thank you.
I saw that Samuel raised your hand, but I think it was the same question as Patrick, right?
Absolutely.
Yeah, good. So Francisco, you can go on.
Hi, thank you very much for the presentation. Very interesting. I have a couple of questions. One is this, which is the diameter of the spot that is receiving the retina, and if you treat only for example, in the GA, which part of the retina you treat? Only the damaged, or do you go beyond, or if you treat the whole eye? And the second question is, how long does the treatment takes if it's painful? And I'm assuming that you only need dilation drops and anesthetic for the patient.
Yeah, the first question, how large is the spot? I mentioned 2 to 5 millimeters.
2 to 5. Okay.
Yeah, it's relatively large spots. The industry have been trying all kind of tricks to solve the efficacy and safety problem before. So there are modalities like micropulse lasers. There's a 5% duty cycle. You do extremely small spots, but the field has been moving to the direction that you can actually do a huge number of these spots. So the overall result is almost the same as using one large spot. And our modality, it's kind of step back to, I would like to say 10 years back. This was the kind of thinking, but for us, only important thing is the heat shock reaction and targeting these mechanism. I mean, this mechanism of action of having induction of heat shock proteins, activation of autophagy, and activation of antioxidant systems. So the modality itself, it doesn't really kind of. We have moved from this laser titration field and other non-damaging laser field to kind of applying our own method. Yeah, your second question, how long does it take? Current protocol, there's a 30 seconds calibration period, and after that 60 seconds treatment, and that's it. And we don't expect it to be painful because it's a sub-threshold treatment. Of course, there are bright lights, so a little bit uncomfort, but nothing painful.
It's bright light, but it's infrared. You mentioned 800 nanometers.
Yes,
so.
yes, but for this photonics part, and kind of core of the innovation is get the temperature exactly from the treatment spot, nowhere else. So that requires other light sources, and it's pretty complicated optics system. So there are other bright lights used.
Patrick.
Thank you.
Yeah, one follow-up question. If you're related to the spot size and where you apply, do you have additional imaging modalities that help you identify where there are critical areas, let's say GA areas in the retina, or is it just that you kind of more holistically treat the whole area?
No, that's diagnostics is done before the treatment, and then the retina specialist chooses the treatment area and uses our device, but there's no diagnostics in our device. There's imaging, of course. We have 3 cameras involved, but typically in retinal treatment devices, the imaging quality is not in the same level as diagnostics. Now, this is our second version of the device, so maybe in the future it will include the same level of imaging quality than the best possible diagnostics companies provide.
I have a final question, Yanni, about the eye tracking. I don't know if you're using the eye tracking that is similar in other applications, or do you need something that is specifically developed for this application in particular?
We are currently using a commercial camera system from the industry, but also developing our own technologies. It's retina imaging is not difficult, but it also gets complicated when you have so many light sources and kind of reflections and everything needs to be taken care of. But pupil tracking is the most important one in the non-contact laser system. Eye movements, small eye movements are okay, but the larger ones needs to be taken care of. So in this field, if there's in the audience some eye tracking people, you welcome any contacts.
Mm-hmm.
But when we did the kind of research on the commercial side, there are only a few companies that are able to provide fast enough systems to be used in our system, our application. Okay,
well, it is an open question for the audience. You can contact us later on if somebody's interested in further discussion. Okay. So thank you again, Yanni, for the time. Thank you for your presentation today. For me, it's a pleasure now to introduce our next speaker. So always a pleasure to see Samuel Bucor in the room, CEO of the company Imaginize, and he's going to present adaptive optics retinal imaging for early diagnosis and faster drug development. So Samuel, now you are not member of the board of EPIC, so I hope you will have also time to join more our online meetings with presentations.
Absolutely. Thank you very much, Antonio. Well, thank you. I hope that you are able to see my
everything good.
Slide. Okay, perfect. Okay. So I will make a short intro of Imaginize, and I will explain what is adaptive optics for those who don't know this technology, and how useful is it in retinal imaging. And I will describe a little bit 2 systems that we have developed. One is very mature. This is the adaptive optics fundus camera, which is on the market since more than 10 years. and then multimodal and multi-scale retinal imaging device, which is at a prototype level, but already 3 or 4 in some location. And I will conclude. So a few words on Imaginize. We are located in Orsay, close to Paris. We built the company in 2003. So there is a whole story, not always in retinal imaging, but in aberrometry in the first part of the life of the company. So we learn a lot how to measure very well an eyes, and we had some competencies in adaptive optics. So we developed an adaptive optics imaging device. Mission is really to save sight, to make the best contribution as possible to save sight and even to save life by providing extremely accurate information on the retina, which are either retina health information, but more global health information in the retina. Because you can see vascular details in the retina, you can see, you can detect some hypertension parameters or things like that. So it really can go far. We believe that this technology that I'm going to show you there is a new gold standard, can become a new gold standard in retinal imaging. We are a small company, you see 20 people, 3 million of sales, but we have a strategic partnership with Imagine Optic. I'm also the co-founder of Imagine Optic. So I want to say a few words on Imagine Optic in 30 seconds. So Imagine Optic was built in 96, so nearly 30 years ago, and we are focused on wavefront sensing techniques, especially Shack-Hartmann techniques, and adaptive optics solutions. So there is some sensors, some deformable mirrors, systems based from optical metrology, and various kind of system for adaptive optics solution. Imagine Optic is not working in ophthalmology because we have this daughter company, which is Imaginize, working on this topic. So the total group at the end of the day, it's about 100 people working on this topic. What is adaptive optics? So it was introduced by astrophysicists who wants to look at the sky, the deep sky, and what happens is that because even with big telescope you are traveling through atmosphere, the image is blurred, and is blurred because of wind in high altitude, and the wind is going to introduce aberration on the optical path between the star or the group of stars you want to see and the detector. So if you introduce adaptive optics, that I will just behind, you are able to improve the image drastically. You can have some very good details. This is the center of the galaxy, and I will show another image in the next slide on that. So you can see there is a real improvement of the image, and this is exactly what we are going to apply in the eye. So here is the basic principle. The distorted wavefront is arriving in a system, and it's going to be measured by a wavefront sensor, but on the optical path, you are able to place a deformable mirror. Deformable mirror is a membrane that can be addressed by pistons, by actuators locally, and then can change of shape locally. So you can have a shape which is exactly the inverted shape of the aberration of the incident aberrations. And then you can correct that. So here you can see there is a wavefront with some astigmatism, which is a classical aberration due to the atmosphere, which is going very fast. And then at the end of the day, you have a good wavefront, quite uniform in color. So that means a few nanometers or tens of nanometers of aberration instead of a few hundred of nanometers, even a few microns of aberration here. You can see the same kind of image. This is the galactic center. And if you have a classical telescope, you see just light. And if you use adaptive optics, this is what I've done. Andrea Guess in 2020. Well, she was Nobel Prize in 2020, and then she were able to see the stars that are at the center of the galaxy, and she observed that the star at the center of the galaxy have a specific movement. This is how she detects that there were some massive holes at the center of the galaxy. So we are going to apply this to the retina. If you look at a retina with a classical device, typically the resolution, the spatial resolution is about 20 microns. So when you zoom, you can see this kind of image with some shadows of vessels, maybe some small structure that you can try to imagine. Then if you are able, so this is not exactly the same as in the atmosphere, but the light which is going in your eyes is going to be distorted by the cornea and by the crystalline, which has some aberration, which are not good optics, and which are changing from one person to another one. So it's quite static aberration. You don't need to go very, very fast, but then you have to correct that in order to get this kind of image. This is all the spots that you can see here are cones. So you're able to see dynamically. This is a live image of the retina. It's a quite small field, of course, but you can see really microstructure, micro details in the retina. You can even make some segmentation to count the cells, to count the neighbors of the cells. This is some kind of biomarkers. I will make a list of biomarkers that we have in hand, thanks to this system. So there is a lot of application, and I was saying in my title, early detection. Yes, early detection, because you can see some details at the cellular level. So then if there is some change in the structure, because of the higher resolution and the higher level of details, you can have some very smart detection of change. You are able to monitor some therapy. You are able to evaluate the progression of a disease in the eyes after just a few weeks. I will show some image on that a little bit later. So we have developed two devices. The first one, more than 10 years ago, it's AO fundus camera, classical fundus camera, and the RTX1. It is a certified system. It's very easy to use. Adaptive optics is, there is a reputation of that adaptive optics, it's a difficult technology to handle. But here, in that case, ophthalmic doctors are just placing the patient in front of it, and they make the acquisition, and everything is completely fully automatic. And in the same way, we have done now a new device, which is at a prototype level for research use only. And this one is a multimodal, multi-scale, and I will detail it a little bit later. Now, just focusing on the RTX1. So you can see, oops, sorry, up. So you can see the standard image. You already seen one. We can add a module to change the lighting. So you can see some pigment epithelium and RPE cells. So it's a different layer. You can add some other modalities for research, especially for research, that allow to make lots of papers. So we have lots of scientific papers, more than 250 papers. And then this device is also now used because it offers lots of possibilities in new biomarkers. We have developed some tools for imaging protocols to make imaging protocols in case of clinical study or clinical trials. So more than 250 publications, more than 20 countries for more than 100 units already sold worldwide, especially in Europe, but also in the US. In Europe, it is certified, so there is no need of specific legal process. In the US, you need to get an IRB. And I could add on this slide that there is, in adaptive optics, which is a quite new technology, there is a little bit more than 50 or 55 studies right now on clinicaltrials.gov And you can check that there is more than half of them that are done with this device. A lot of them are done at the academic level, but more than half of the clinical trials are made with this device. You can see Michel Pack is someone who adopted the technology very early, and lots of papers are on inherited retinal disease, but also on AMD. Well, there is a lot of application. The list of AO biomarkers that we have been able to demonstrate and to work on, and we're still working on in clinical trials, and more especially one that could interest Gianni on atrophic AMD. We are able to measure short-term general atrophy, geographic atrophy, progression. Here you can see there is two image. Okay, you can see that. So this movie on the right, each image is between 2 weeks and 1 month change. So you are not obliged to wait for 6 months before seeing the change in the eyes and evaluating the damage of the disease. But every 2 weeks, you can have an image and see the progression of a few range of cones that are disappearing. This is the lesion is in that area, and this is the safe area, but you see that things are moving. There is some dark points that are kind of proteins, pack of proteins that are changing also of place. So very interesting way of seeing the atrophic lesion, how it change. And of course, because it is very fast, then you can see how a treatment can be efficient in a few months instead of a few years. So you can save pharma companies, therapeutic companies can save a lot of money in the clinical trials thanks to that evaluation. Now we're checking just one slide on N2 OCT, which is a new device supported by Europe. We have a lot of support from Europe since a long time. So you can see all the modalities. We have, in that case, large field modality with SLO, OCT, OCTA. And then once you're able to check the location of interest, then you have the zoom with the adaptive optics modality, the adaptive technology which enhance the modality. So you can get high resolution in IOSLO in face vue. Then you can have the high resolution, cellular resolution in AOCT, and you have a very fine description of vessels in AOCTA. Some reference paper indicated here. You will have the copy of the slide, of course. And then, so we are working right now with Netherlands group to introduce AI in image analysis in order to make some evaluation of very early capillary damage or damage in the nerve fiber for glaucoma evaluation and glaucoma progression of the disease. So very, very promising device to provide a lot of information. Just to summarize or to open the discussion, but so adaptive optics is not very known right now in that field, but this is, you have really the most advanced industrial adaptive optics retinal imaging system with the RTX1 that I just introduced. We can improve all kind of existing modalities, even on specific development. The higher resolution provides earlier diagnosis. The highest resolution provides faster and less expensive drugs development. And my conclusion is that we believe really that adaptive optics is a game changer, not only in the ophthalmology field, but also for global health and wellness. And why I say wellness, just I can explain, because we start to work on neurodegenerative disease, and we have some intuition, let's say intuition right now, because there is not yet the demonstration, but that we can detect probably 20 or 30 years before Alzheimer context in the retina. And so that means wellness, because then the people and the family can be prepared to this disease. And of course, if tomorrow there is some treatment for that, the people can apply this treatment, at least in the food or in some other process to decrease the speed of progression of Alzheimer's disease. And so the EPIC questions. So we're setting the full device or some of the components. We are open. The components are integrated in other technology. We are offering a unique expertise to reach the retina with a cellular resolution. That means if you apply a laser, so not sure that it is interesting for Gianni, but if you want to make some surgery, you can reach the retina with the smallest resolution as possible in order to preserve the health of healthy cells inside. And what we are looking for, just in the industrialization of the new device, the prototype, the multi-scale and prototype, the M2 OCT, we are looking for swept source. So we are working right now with a 200 kilohertz swept source, but probably we have to decrease a little bit this performance to make it more affordable for the market. We are looking for low-cost SLED in that range, a few milliwatts, not critical, but really low cost. And in human resource, we are looking for a clinical application specialist. If you have this in your network, don't hesitate to transfer the information. Thank you very much.
Thank you very much, Samuel, for the presentation and also for the EPIC questions. So we have now also for time for Q&A. You already answered a little bit one of the questions I had, and it was the use of the retina to go into these neurological diseases. I remember one presentation we had last year in Barcelona using Raman spectroscopy, connecting a spectrometer to a modified SLO to make some Raman measurements. Do you think could be combined with your technology to get better results?
Yes, I can. I think that, well, in all the cases that we have studied, you can always cumulate the advantage of a new modality and adaptive optics.
Mm-hmm.
Most of the time, you can cumulate advantage, especially in terms of spatial resolution, or sometimes, especially in microscopy, you can go deeper in the tissue,
Mm-hmm.
thanks to adaptive optics and with special algorithm, of course. But yes.
Okay, I will share later some information with you because perhaps you could have a look to see if there is potential collaboration.
Okay, great. Okay, thanks.
And I also had a question regarding the potential users of your solutions. You mentioned the pharma company that can perhaps test the drugs and also the clinicians. Are you going exploring both options? Do you think for the future you will go in one direction more preferably?
The reality is that pharma are not the direct customer.
Okay.
Because they never invest in some device, or it's very difficult for them to invest in device. Anyway, when they do clinical trials, they don't want to get directly the device. In general, they are using a CRO. The CRO is going to rent the device during 1 year, 2 years, 3 years, depending of the size, the dimension of the clinical trials. But so that means we are working more especially with clinical centers that are able to participate to clinical trials, and there is a network of this. So
yeah,
it's true that what I can say for our markets. So the RTX1 is just a fundus camera, improved fundus camera, and we have a few routines with it, but we are not able to reach a mass market with this device. It's not enough. The functionality is not enough large, very accurate, so very interesting for some people doing some research, but not enough large in terms of functionalities. But the M2 OCT is one OCT. So the market which we are targeting is OCT market.
Yeah.
And in this OCT market, then we will have the high-end OCT market with this device, because we will be able to provide really high resolution.
Yeah. Good. I have a question from Yanni. Yanni, please.
Yeah, hi. Interesting technology. Regarding the clinical trials, we've been kind of trying to find the publications related to kind of speed of geographic atrophy growth. Have you faced any studies that would have a kind of classified the patients to different groups, or?
I'm not sure of that. I'm not directly involved, but I can send you a clinical paper summary about AMD that we have published a few months ago. I will check the information which is inside, and I will send it to you.
Okay.
But there is some current clinical trials. I don't have the latest results on that.
Okay, thanks.
Patrick.
Yeah, thank you. Really interesting technology, particularly with the new device combining, SLO and then OCT and OCTA with its AO variants. I think this is really interesting. In this device, can you comment a little bit on the field of view of the different modalities? Because I would assume an SLO has fairly large, and then it scales down. I was interested on how this scales, so to say.
Yes, so for each time you introduce adaptive optics, you are reducing the field at plus minus 2 degrees, something like this. So it's quite small. But the large field, SLO, OCT, and OCTA, is a quite standard fields. I cannot give you the exact value right now, but it's not like Optos device, Optos Nikon device, and not very ultra large field, but it's a large field. I'm just answering to the standard in the OCT field. Just what I can say right now, but the same. I can send you the data sheet of the device, and everything is inside.
Is the idea then also to kind of stitch together perhaps a different, AO regions to have a larger field?
Can you repeat your question?
Is the idea, or is it possible to stitch together certain fields and?
Yes, this is what I was showing in one image for the RTX one, which is only a small field. And then we have a software collaboration with a software company to make montage with all the image, the number of image you want, so with a small recovering place. But in the M2 OCT, you have the large field, and it is already inside the machine. It is coupled, so that means even with your hand, you say, I want to see here with high accuracy, and then it will switch directly on adaptive optics at the good location. And after that, the other question, which is going in the same kind of problematics, from one visit to another visit, we have the location of the zone, but the location is not enough accurate, so we have the possibility to make a very good trim, very good superimposition of image taken from one time to another time, less than one cell of recalage trimming.
Thank you.
So if there is no more question, thank you again, Samuel, for this presentation this afternoon. Interesting technology, and we keep an eye to this multimodal system. So best of luck with this.
Thank you very much.
And well, we had a really couple hours of presentations, and now we are going to another very interesting topic that is optical modeling. So it's a pleasure for us to have Sila Timarfulep from the Optical Photonics Workshop at Ansys, and she's going to present the optical modeling of human eye for ophthalmologic industry, then also vision correction design. So Sila, the floor is yours.
Thank you. Yeah, welcome everyone, and thanks for joining us today. My name is Cilla Timarfulep, and I'm the application engineering team lead for the Optics Photonics workflow team here at Ansys. In this presentation, I will talk about how to model the human eye using the Ansys optics tools. So first, I will discuss the motivations. Why is it important to do numerical simulations and what's their benefit for us? Then I will deep dive into a specific application of designing a multifocal contact lens for presbyopia correction using the Ansys optics tools, namely ZMAX Optics Studio and SPIOS. Finally, I will summarize the main points and draw some conclusions and open the discussion. So the human eye is our most important sensory organ. It conveys more impressions than any other sensors. And also there's a significant portion of the population which suffers from different types of vision loss. This means that there's a huge demand for better understanding of the physiology and also the anatomy of the human eye. And one way to gain that better understanding is through numerical simulations. In that case, our goal is to mathematically represent the optical components of the human eye. And due to the large complexity of the eye itself, there's a large variation in the model complexity which exists in the literature. There's models ranging from simple paraxial ones, including just a couple of surfaces, to really complex models, including aspheric surfaces, as well as gradient index materials. But there's no model which fits to every purpose or fits to every eye. Rather, the model needs to be fitted to the simulation task, which in fact can be quite diverse. The applications of numeric simulations include exploring the retinal image quality under different conditions, also predicting the vision quality, and analyzing the effect of ocular biometry on the imaging quality itself. Besides, numerical simulations can be used to design ophthalmic instruments for diagnostics, for example, wavefront sensors or OCT devices, as well as for designing corrections for patients with vision loss that includes cataract patients and intraocular lens design for them, and eyeglasses or contact lens design for presbyopia patients. In the specific case of presbyopia, the eye loses its ability to focus to close object distances due to aging. What that means is that patients need a good way to restore the vision quality at a wide object distance range. In the recent years, there's been increased interest in simultaneous vision multifocal contact lenses as the preferred method of correction due to the demanding visual area and their cosmetic advantages over spectacles, as well as the increased practitioner confidence to fit these lenses. But what we need for that is numerical simulations again. So the Ansys Optics portfolio provides multi-scale and multi-physics simulation platform ranging from nanoscale optics to system-level simulations. On the nanoscale level, we have Ansys Lumerical, which performs full-wave electromagnetic simulations to design diffractive optical elements, metalenses, waveguides, as well as to simulate emissive and absorptive structures. On the macroscale level, we have Ansys ZMAX Optics Studio for optical design and analysis, optimization, design validation, including optomechanical tolerances as well. Finally, on the system level, we have Ansys SPIOS, which enables 3D environmental modeling in a native CAD environment, including different light sources and various sensors in order to model human vision and describe and visualize how humans or machine vision systems would see a specific image. What this means in the specific application of correction design for presbyopia, we can leverage the Ansys Optics tools with the following simulation workflow. We can start by modeling the human eye itself in ZMAX Optics Studio's sequential mode by ray tracing. Then we can leverage the powerful optimization capabilities to design the contact lens correction for the patient. Once the design is ready, we can take advantage of the optical design exchange capabilities within the simulation software and use Ansys PIOS for a full system-level analysis, including intraocular scattering as well. So to deep dive into this workflow, we can start the modeling in Zemax Optics Studio and start with the gold standard paraxial Gallstrand exact number 1 model. This model represents the crystalline lens as cortex and the nucleus, and it only uses spherical surfaces. But in order to accurately describe all the higher order monochromatic aberrations of the eye, we need to introduce aspheric surfaces into the model. Furthermore, in order to describe the significant longitudinal chromatic aberration of the eye as well, we introduce custom materials to describe the chromatic dispersion of the media. With this, we can achieve a very good representation of the unaccommodated average human eye. What that means is that it also provides a good representation of the presbyopic eye, which has lost its ability to focus to close object distances. So we can set up a multi-configuration system and change the object distance in order to analyze what the patient would see at different object distances. As we can see, the patient would have a quite good vision at far and intermediate distances. However, the image becomes completely unrecognizable for close reading object distance. So in order to restore the vision quality at a wide range, we design a multifocal contact lens, which consists of 3 concentric zones, starting with a central spherical zone for near vision, then an aspheric transitional zone for intermediate distances, and finally a peripheral zone, spherical again, for distant vision. This type of contact lens is a simultaneous vision multifocal lens, which means that the patient sees all the focused and out of focused images at the same time, and the brain learns to post-process these images. What this means is that from the design point of view, while the zones are used all at the same time, they have to be optimized separately for the specific viewing distance. So to achieve this, we can set up variables in our model, the radii and conic constant of the different zones, and we can target our optimization to achieve the smallest RMS spot size, which can be a default metric function in Optics Studio. As we can see on the images, with this contact lens, we can trade off some far vision to increase the vision quality at close object distances, and therefore balance vision quality in a wide object distance range. Once the model is ready, we can leverage the easy and accurate file exchange between the Ansys Optics tools and create a full system visualization and performance analysis in SPIOS. So this means that we can set up a realistic vision test scenario, just as in the clinical trials, use the standard Snellen chart, take into account the sensitivity spectrum of the human eye, and customize our detectors to best mimic the resolution and the curvature of the retina. In order to take the model further, we can also introduce intraocular scattering in the model. Based on the literature, the most important sources of scattering in the eye are volume scattering within the lens and the cornea, as well as surface backscattering from the retina. To start with the most prominent one, we can introduce volume scattering inside the materials. We can do that by defining the protein molecules which cause the scattering effect, and describe the dominating mean scattering, and customizing the model by defining the radius, the density distribution, and the refractive index of those particles. With this, we can model different scenarios, such as a normal aging crystalline lens, or a crystalline lens with a cataract affected. So by numeric simulations, we can visualize and evaluate, assess the performance of different scenarios. We can see what the presbyopic patient would see at different object distances, how the image quality worsens as we move the object closer to the eye. Then we can analyze how the designed contact lens would correct the vision and balance the quality between the different viewing distances. We can also see how cataract would make the image much worse, requiring for a different type of different type of correction, such as replacing the opaque crystalline lens due to the high amount of scattering by a new artificial intraocular lens. So to sum up, we have seen how to use the Ansys optics tools for creating an efficient simulation workflow for modeling the human eye and analyzing vision quality and evaluating it under different conditions. We can leverage the powerful ray tracing and optimization capabilities of ZMAX Optics Studio and take advantage of the system-level performance analysis and visualization of SPIOS. What this means is that lens manufacturers can analyze conditions and predict the vision quality in a much broader range of scenarios than it could have been done without simulations, which can lead to improving the design conditions and achieving much better correction designs, which leads to increased patient satisfaction. On the other hand, we can also use similar eye models for intraocular lens design for cataract patients. And this means that designers can optimize intraocular lenses and evaluate the postoperative vision quality even before the surgery by numeric simulations. If we take into account ocular biometry as well and customize the models based on the measurement details, then we can create personalized eye models, and that enables us to design customized correction for the given patient. Also, these eye models can be used for virtual prototyping, which means that we can leverage the models during ophthalmic instrument design, so to visualize and evaluate what the doctors would see during the actual clinical trials without building the devices and only using numeric simulations. And to close the topic, what you can do for us and what we can do for you, we develop optical simulation software. So we would like to help people to perform numeric simulations, to improve the research and development by virtual prototyping and increasing the scenarios that you can discover without building prototypes. This means that you can speed up the time to market of the devices. And what we would like to get from you is that we would very much like to hear what specific scenarios you would like to model, what are the challenges that you face during numerical simulations, and if there's any special tools or requirements for the simulations that you would need so that we can customize our product development and take that into account and introduce it in the future. Thank you very much for your attention.
Thank you very much, Sila. So we have time for questions. Yeah, Francisco?
I was preparing my finger to be the first one on doing the questions because we can stay here forever. We can actually I reached you out a few months ago on this topic, if you remember.
Yes, exactly.
Well, I was not. I haven't started working with SPIOs, but the simulating intraocular scattering, in particular with diffractive IOLs, looks like a promising scenario. and we can actually, I would love to work with it on it together, because it's not just, I mean, it's not only the scattering, is that the lens will be tilted or decentered when they are actually usually designed to be performing properly centered and properly tilted. And I'm wondering if, how do you, if with that software, how do you estimate the visual acuity?
So there's a couple of capabilities to post-process the images that are required by ray tracing, and in that post-processing we can include different types of analysis. One can customize a model which introduces neurophysiological post-processing that's done in the optical nerves and in the human eye. So yeah, by programming capabilities which we offer, there's infinite possibilities to customize the post-processing of the imaging. And once we simulate what's on the retina, then we can go further from there to estimate visual acuity as well.
Awesome. Thank you. I will stop here and I will continue later with you if you want.
Yeah, of course.
Very, very interesting. Thanks very much.
We have time for more questions. If not, I can leave Francisco to have another one. I have one specific question, because you show in the intraocular scattering model that you have some specific values of radius and particle density for the cataract and for the aging, but it's something that the user can change a little bit in order to make kind of evolution, or is something that for the moment is fixed just to see the difference between the two situations?
Oh, no, it can be customized as the user wants. So these are just two specific values that we used for demonstrating what would be the case for like a normal aging eye and an eye with a cataract. But this can be customized based on measurement data or to describe how the cataract evolves within the eye. So this is absolutely customizable. It can be changed. Yeah.
Okay, good.
So yeah, these are parameters that can be changed. Yeah.
Yeah. I don't know if there is any other question from the audience. You can make another one, Francisco, if you want.
Absolutely. Thanks for pushing me. Actually, following your question, very interesting as well. It will be, I mean, I'm assuming that when you are doing this simulation of the cataractous eye, you are applying the same density of scattering or the same amount of scattering to the whole lens. It will be interesting as well to see which are the differences between a nuclear cataract, posterior cataract, a cortical cataract, because from the clinical practice, we know that the posterior cataracts impact a lot the visual acuity as they are closer to the nodal points of the crystalline lens and so on. It's really promising if we can, and it can be a very useful tool for clinicians to show the patients what is happening on their vision.
Yeah, that's perfectly possible to like separate different parts of the crystalline lens and set up a different scattering model within the different parts to mimic what's happening inside the eye. Yeah, that's the details that can go further and further. That's what I meant by saying that there's no model which fits for every purpose and every eye. It can be customized based on the application, based on the patient. Yeah, there's a lot of room for taking into account more and more details and refining the model based on that.
Good. So thank you very much, Hila. Thank you again for your flexibility being here this afternoon.
Thank you very much.
And for sharing all this valuable information. We can move to our next speaker, that is Francisco, in fact. So it's a pleasure to have you here, Principal Optical Engineer at the company Bausch Lomb. and he's going to present the work by rehabilitating intraocular lenses performance through simulated BI. So Francisco, the floor is yours.
Can you see my screen?
I think now, it's good. Perfect.
Here we go. So actually following the former presentation, what we know, and this is why my questions were going through how to simulate visual acuity. What we know in the clinical practice with the patients, the way we address the visual acuity of the patient is through looking at a chart that have some letters that the patient have to say, have to identify. There is a very almost new approach to measure the performance of the lenses, and this applies to contacts and to intraocular lenses as well, that we want to know the defocus curve. That is, how is the visual acuity as a function of the distance or a function of the defocus. And the way the clinicians make it is inducing lenses, adding lenses in the phoropter for each defocus and remeasuring again the visual acuity. When we go to simulations and to optical bench measurements, we cannot simulate that directly. What we do is to measure the quality of the lens that is usually represented. Here we go. It's usually represented with the MTF that represents the optical quality for a single defocus. And what we can do is to measure the MTF at a single frequencies for all the defocus to know which is the optical performance. Unfortunately, this straightforward measurement is not correlated with the clinical visual acuity. And it doesn't go to the next slide. Here we go. So the next step is actually, and there are a few literature on this, that is measuring the area under the curve between a certain number of frequencies and to perform a correlation between the clinical data and the area under the curve to get the expected visual acuity. So there are two main questions, that is, which is the range of frequencies we should be using and which is the equation that better correlates with the expected visual acuity. So in a first step of this work, what we did is to simulate with ZMAX a modified Liu-Riemann model I with a corneal spherical aberration that mimics the average corneal aberration of the patients using white light and simulated with a Envista monofocal lens, that is the Bausch Lomb monofocal, that we know pretty well the clinical data. So, and we matched this, the process of the calculating the area under the curve of the MTF with the data of 445 eyes coming from two different clinical studies. Here are the first, there is a lot of equation, a lot of numbers here, but basically is that what I want to show is first that the results of using area under the curve between 50 and 100 are very similar. Second, that after, besides logarithmic and linear equations that do not fit pretty well, and we can see that the correlation, the R square is not very high. Anyhow, logarithmic is 0.95, that is pretty high. The best correlations, we got it with exponential, logistic, and rational approaches in both 100 and using 0 to 100 cycles per millimeter and 0 to 50 cycles per millimeter. The power law equation did not offer good correlations as well. We can show this as well with using this plot where we see how close are the fitting to in both with both approaches. For simplicity, we decided for the second step of this study to get this equation, this exponential equation with the frequencies from 0 to 100. In a second step, we wanted to know which was the expected performance of a new, it's called monofocal, enhanced monofocal lenses. That are lenses, these are monofocal lenses, but are designed to give a little bit of intermediate vision. But what we wanted to know is not only how much is this intermediate, we are really interested on knowing which is the robustness or the variability of these two designs when we use different amounts of spherical aberration in the cornea, some corneal astigmatism, some IOL decentration, and some IOL tilt. All these values are inside the limits of normal eyes. So if there are no pathological eyes included in this, so we can induce this stress in even a larger amount of patients if we go to pathological corneas or pathological eyes. Surprisingly, what we found is that the impact of astigmatism is very similar in both designs, and decentration and tilt, they are very similar as well. So the differences are very small, but and actually they have very low impact on the performance. So we can say that the lens perform pretty well, no matter if they are which are the tilt, which is surprising, because actually tilt and decentration, where residual astigmatism is measured in the clinical practice, decentration and tilt is not that usual. Nowadays, we have new generation OCTs that can measure which is the actual decentration and tilt of the IOL, and we were expecting having more variability or a higher changing impact in the performance when tilted on decentration, but it resulted to be that with these lenses the impact is very low. What was really surprising is that when we changed the conic constant to induce several amount of spherical aberration on the IOLs, we observed that the area of variability, that is the area between the extremes of the spherical aberration between 0 and 0.55, is higher with the standard monofocal than with the new monofocal or enhanced monofocal here. And we can see as well that there is a shift of around half a diopter when we move from spherical aberration neutral to a corneal aberration, a higher aberrated cornea. That happens with both lenses, this shift, which take us to the following takeaways. That is that the Aspire lens, that is the monofocal plus, seems to have greater resilience to variations in corneal spherical aberration compared with the standard monofocal. The impact of tilt and decentration is very small to both lenses, and the impact in astigmatism is very similar to both lenses. And that the spherical aberration should be, or the conic constant should be included to, for example, when we calculate the IOL power of the lens, because it's something that we actually measure in clinical practice, but we are not used to take into account it in the IOL calculation formula. So this point can be very can be nice for further generation of IOL calculation formulas. What can we do for you? Well, Bausch Lomb offers a very large ophthalmic portfolio across different business units, collaborating in next generation product development, integrating optical modeling and clinical needs is possible, and we can act actually as potential bet to test translational results with our platforms. And what can you do for us? Providing new measurement technologies that can improve the accuracy on how we measure high-order aberrations of the cornea, how we measure tilt decentration, the effective lens position, everything related with the physiology of the eye will be really interesting. Refining diagnostic to improve IOL selection and personalization, and we can co-develop data-driven tools to improve the patient satisfaction and the surgical outcomes, and as well the surgeon satisfaction. Happy patient, happy surgeon. That's all from my side. I'm gonna stop sharing to see your faces, and if you have.
Thank you very much, Francisco. Yeah, as Francisco mentioned, it's time for questions, so we can make some questions to him. I could start with a question about the visual simulators that some companies are developing nowadays. How do you see them as a potential support in this kind of perhaps refining diagnosis and helping in the developing of IOL designs?
Very good question. It's promising, especially well, depending on the design of the lens and the optical principles, they can have some differences between the real performance and the simulated performance. But for sure, they are really helpful to have an idea on how potentially a new design can be better or not to a former design, and to have an idea to how which of the features of the new lenses can be beating the former designs or competitive designs. In contact lenses is really helpful because it can gives you a very quick address of which is more comfortable to the patient from a visual perception standpoint. Contact lenses have the advantage that you put the lens and you can take them out. In intraocular lens designs, that's more complicated. So this visual, anything that you can simulate and show the patient which could be the potential benefit of one design among the other is welcomed. There is in cataract in particular, there is still big improvement that can be done, or that should be researched. That is that we still cannot see through the cataract. So if you are using a simulator that doesn't correct the effect of the scattering of the cataract, it will be not as nice as being as, for example, in the contact where there is no additional scattering, that you are not removing any kind of scattering with the treatment. So
yeah,
promising, and actually I'm pretty sure that they're moving forward, and the field and there is open room from these technologies to spread.
And can you explain a little bit the process of developing the lens, because you can work in the design and develop a little bit. Is something that Bausch Lomb also proceed by internally to manufacture the lens itself? Is something that you contact with other companies to manufacture the lens itself to then supply them to the clinicians? I don't know if you can tell us a little bit of the process.
Well, Bausch Lomb is, besides that it's a huge company, it's not as big as the main two competitors, but usually we get an optical design that can be done using ZMAX or other tools, and with that design, we stress the design from a design standpoint and make the lens behave as we want theoretically. Then we have to put that lens into the manufacturing process. The manufacturing process is really complicated. It's very, there are many phases to get the final product done. So when we got the final product, we have to measure that the optical properties as was we expected, because what we cannot do, and there is no technology. There are technologies, but not as good as enough to ensure that what we have manufactured physically is the same that what we design.
Yeah.
Among others, lenses go through a polish process. So if you are manufacturing very complex or diffractive steps, you don't know how much of these diffractive steps are being lost through the manufacturing process. But the only thing you can do is to ensure that the optical quality after the manufacturing process is what you expected.
And this is on bench. Is it not on
this
the
is
lab?
on bench. Yes, yes, this is on bench. We manufacture the lens and then do an in vitro testing of the properties to check that the optical quality we expected is similar to the optical quality we are providing. If not, we have to tweak the manufacturing process or to tweak the design to get the results we are looking for. After that, regulatory path and commercialization. That is as well a very complicated process because it's an implantable. So it's a 2B or 3 class 2B or class 3 device that usually requires marketing studies. So,
and for the different designs, do you need to follow different regulations, or You can use
They
one
were-
of the previews that you.
Yeah, depending on the category of the lens, the regulation asks you to test and perform differently. So it's not exactly the same from a clinical R&D perspective, from a clinical design perspective. If you are trying to provide the patient distance and near vision, you have to test distance and near vision. In a monofocal, that's not mandatory. And actually, FDA and medical device regulation are asking you to provide the defocus curve. That is very time consuming, and the patient's fatigue increases after that. So there are things to take into account there as well.
Okay, very good. That's a good summary. I don't know if there is any other question in the room for Francisco. If not, thank you very much again for sharing your ideas and your work today, this afternoon. Thank you.
Thank you. Thank you.
And we
Thanks for the
continue.
invitation.
Yeah, we continue with our last presentation for this afternoon. So we were talking about intraocular lenses design, and we can move into the fabrication process. And it's a pleasure to introduce Altan Yildirim, the CEO of the company Lumos Laser. They develop ultra-fast lasers, and they have expertise in precision processing of intraocular lenses. So Altan is going to present the work that they develop in the material processing in this particular application. The floor is yours, Altan.
Okay, thank you, Antonio, for the introduction. I guess I'm successful with sharing. Yes.
Good.
So hello, everyone. Good afternoon. This is Altan. I am the CEO of Lumos Laser. We are a 10 years of startup company from Istanbul. Actually originated from a university in Ankara, Bilkent University. And we are producing next generation femtosecond lasers for precision micromachining. We have our own invention, a gigahertz rate process in our lasers. And I want to talk today about this gigahertz rate operation, as well as a very interesting application in ophthalmology of our lasers. That's the intraocular lens processing. So femtosecond lasers are applied since 20 years in the industry. They have many different applications. It's really difficult to focus on one field because there are really different lasers good for very different applications. However, our focus is on precision manufacturing and specifically on medical devices, aerospace, and glass processing. However, there are really interesting applications also coming for the real future tech, such as photonic ICs and even quantum technology, as well as in healthcare and bioimaging and spectroscopy. Since our lasers use very low, very short, ultra short pulses, and they can have really low power, and they can interact with any molecules quite easily. So femtosecond lasers are really state-of-the-art tools for precision manufacturing. They use ultra short pulses, hundreds or tens of femtoseconds. So they don't heat the surface, heat the material that they are machining. And this helps to get a single process production, which means we don't heat, melt, or we don't deform the material that we are processing, or we don't leave any residues, or not any, but many residues, let's say, on the material. And it can machine all kinds of materials like diamond, very hard metals, polymers, ceramics, composites, whatever it is. So it really sublimates the material on the specific focus. So it can be really a good tool for innovators from all sectors or high-tech sectors. However, there are some drawbacks, mainly the speed. They are quite slow compared to nanosecond lasers or mechanical processes, and the efforts to speed up the lasers increases cost and the industrial adaptability feasibility goes down. So this is our invention, the cool burst technology. The efforts to speed up femtosecond lasers use high energy pulses, but when we increase the energy of pulses, we lose some advantages of femtosecond lasers, and we start to heat the material. And instead of that, we have tried another method. We used low energy pulses at gigahertz frequency. That's the frequency where the material relaxes during the process. So it's really in a deep physics phenomena of machining. So this way, by sending gigahertz frequency bursts, we could machine materials up to 15 times faster, which is really very important for industry, actually. And Lumos Laser has started with this goal to commercialize this technology and build lasers with this technology. So we are really happy to bring this technology to life, to the market. We have made our first sales, and we have many projects going on. And I'm going to talk about one of the very interesting projects today. These are some of the applications. We also develop our lasers on the way. We have our technology roadmap continuously developing, because working in gigahertz region is a really big challenge. So, but until today, we managed to develop many applications such as medical black marking, glass drilling, or coating removal over a tempered glass, which is very challenging, and medical lens processing and some other medical devices we are still working on with our partners. But we are the laser module developer, I have to mention. So coming to ophthalmology, we have a very interesting project with BSY Biotechnology and two universities for this development. BSY Biotechnology is also a Turkish company producing IOLs. They have both all monofocal, bifocal, or trifocal, even toric IOLs in the product range, and they use casting and traditional mechanical machining methods, and they reached to Lumos, and we have started a project like 3 years ago to machine the lenses with laser. What does it bring? It brings really a lot for the producers. As also Francisco told some time ago, it's a really challenging process, because we are dealing with a polymeric hydrogel, which is not a very stable material to handle. So at the moment, in the processing of optical part of the lens, we use a lot of molds, because we have a lot of different patients. So customization has to take place, and it's quite important, obviously. And when we go to laser process, however, we just have one mold, and we make almost all of the rest by laser. So this gives a lot of design freedom and a really good opportunity for customization. And the second advantage is in the cutting of the lens. So we just discussed the optical part processing, and also we have to cut, and this cutting is also very challenging, because it has to be a perfect lens with very sharp edges and like micron precision. So in order to do that in mechanical cutting, there are a lot of different steps to like molding first, and then cooling, cutting, and it's quite a big challenge, because you have to fix the tiny lens on some surfaces. So there are some scraps and operational efficiency goes down a little bit. And if you really can produce in with a high operational efficiency, this is really very helpful for the future innovation. And this is what we try to do in this project. And also laser is a digital production tool. So this is a movie. I will stay here for a while. Here we have transformed a monofocal IOL into a bifocal enhanced depth of focus lens. You can see that it's done in seconds, and no burrs, no cracks, no burnings or no particles. So it was a very successful part. Then we started to develop the trifocal IOL with using Fresnel rings. This is a patented technology of VSY Biotechnology. And we made also our simulations and tried some first trials on glass. And after that, we also managed to produce. Here you see the trials on lens in our production system. And we have developed our process first according to the lens. And we can change this with respect to the lens design. So these are some results I want to show you. We really managed a clean process on micro scale. Of course, there is room for improvement. There will always be room for improvement, I guess, considering IOL processes. But for a research project, it's quite good progress for us. And we have also used the cleaning of lenses after process, and we really got good clean surfaces. And biocompatibility and chemical tests are also all well done. So we are going for clinical studies. And here you can see the movie of the cutting process. I hope you can see the movies as good as I do. So on the right side, you can see some cut samples. Takes place really fast and also clean. However, I cannot show you every detail. It's still under investigation, but the cutting is also extremely important for the lens quality. So, as I said, we are a startup company. We are 3 PhDs and 2 masters. We are in Istanbul at Boğaziçi University and Özyeğin University. We use both facilities as well. We have more than 30 years of ultra-fast laser design experience and also some industrial experience. And we are really looking forward to for R&D collaborations to discover more capabilities of our lasers in different fields. And so if you have any ideas coming to just presenting the technology, please contact us. And actually, that's all from my side. Thank you very much.
Thank you very much, Altan, for the presentation. We have time for questions. Yeah, Francisco.
Yeah, interesting. Is that process, can be that process done directly in the eye? For example, can I transform a monofocal to a bifocal once the lens is implanted in the eye?
I guess cornea is a problem because the laser has to pass very clearly, and it's a 1030 nanometer laser. I have to ask about it. I mean, there are some efforts to do that, but I don't know if they do it with femtosecond lasers. I guess the LASIK world would have an answer for that right away, but I will note that down. I mean,
And another,
it was also a dream for VSY Biotechnology to modify the lens inside the eye. It's extremely important, you know. If something's wrong, you can correct it.
and that was actually my follow-up question. Can you undo? Do you have a Control C
Control
to
C.
undo?
Well, yeah, no, these are, I mean, I guess these are future questions, but these studies should be really, I mean, I also have medical devices background from other fields also, like cochlear implants, and this is as useful as anything for the patient and the doctor. I mean, what you suggest is very important. I guess it will be done, but we have to first manage it on air. So after that, we can work on the eye. Yeah.
Thank you very much. Very interesting.
Thank you.
I have a question for you, Altan, in terms of laser parameters. In this particular application, do you think with the development or the system you have nowadays is what you will use in the future? Do you have in mind any other parameters, change perhaps wavelength, thinking in a different burst frequency? Any ideas for future developments?
Yes, we already work on second harmonic. We have an inquiry as well on the second harmonic generation. And I mean, in principle, yes, we can, but we are a startup and really focused on perfecting this gigahertz rate process. But after that, we are really eager to develop. I mean, our R&D team is much more eager to develop that. So it's I mean, we even have a non-mode lock laser design, femtosecond laser design, which is just waiting for us to have time and to develop. So we already developed it with a project, but we can commercialize it. Then femtosecond lasers will be much more stable.
Yeah.
You know, maybe that. So, and with different wavelengths and different properties are in the pipeline, but there we are following the trends. You know, there are many good femtosecond laser companies. We are also following them. So, but we are the trendsetter in gigahertz burst technology. So we really want to focus on that and advantages, because like photonic integrated circuits also use that gigahertz, and also there is the ultra stable data storage project. Maybe you know, also very interesting. So data won't be stored on semiconductors, but on glass or ceramics. So we are really looking into what we can do.
Yeah, very good.
But medical devices is very interesting. Also, we are really motivated to go on that. We have another project with a dentist working on implants, machining perfect implants with zirconia and so on. Very exciting, but challenging also.
Yeah, of course.
Yeah.
I don't know if there is any other question for Altan. Well, if not, thank you again for a presentation this afternoon showing
Thank you very
our
much.
capabilities. And we will finish. Thank you, saying thank you again to our sponsor today, Tecnottica. So we have Alberto Fasoli that while you are answering the online poll that will help us to improve our next meetings. Alberto, perhaps you can share your screen and tell us a little bit about Tecnottica and your capabilities. And thank you for supporting our online meeting today.
Yes, it's a pleasure for me and for our company, of course. Let me just share my screen. I just have prepared a very short presentation. Okay, since the time is already finished. Okay, can you see it?
Yeah. Going to full mode, I guess.
Yes.
Just to remind in the meantime that we will visit your facilities in September.
Yes, and 9th of September, if I remember correctly.
Correct. We have this meeting in the industrial laser processes in Monza, Vimercate, and we will visit also Tecnottica facilities around there. So looking forward for it.
Okay, so I will be short today since we'll have more time during the visit at our company in September. So as I was telling, thank you again for the sponsorship opportunities. I'm Alberto Fasoli, and I'm the Chief Operating Officer here for Tecnottica. I have joined the company more than 10 years ago in 2013, and my total experience has been made directly on our production line over more than 10 years of direct experience in the optical manufacturing. So Tecnottica is an historical company since it's been founded.
We don't see the full mode, Alberto. I don't know if
you don't see the full one. Sorry. Okay. Let me just
checking that.
Yes, I thought it was already okay, but.
Okay, and now you need to you need to go up to the menu because now we see the two slides, the next one as well.
Yes. Let me just redo it. Okay, because I'm working on two screens. Sorry.
No problem, Alberto.
Okay, I will leave it as it is because it's a bit complicated to switch it right now. Also,
okay,
also if it's a bit small, it's fine. So as I was telling, Tecnottica has been founded in 1957 and has established over the years as an optical manufacturing company, starting working mostly on local market for magnification and lighting application on the industrial field. But in the early 2000s, the company decided to invest into a complete new CNC-assisted line of machineries and a dedicated metrological laboratory, creating at that time a new line of production focused on the manufacturing of high precision optical elements. So in that way, we start work for very different applications, joining automotive sector, medical and biomedical, machine vision, and many others during the years. And also we have started selling all of our products all around the world, and actually more than 50% of our annual revenue coming from the export market. Since we are talking about ophthalmic, our experience in the ophthalmology and ophthalmic application is due to various projects we have joined for these purposes. Of course, I can't be too much explicit talking about some machineries or some projects we have helped build, but just to make some example, we have, for example, more than 15 years production that is still currently active for various optical elements.
You can move, Alberto, sorry for disturbing again.
Yeah.
You can move to the next slide.
Oh, it's just one slide. It's just one slide. It's a very short one, so I will leave it as it is.
We are in the first one yet, in the
okay.
You need to move to the second one.
Okay, you see the second now?
No, we still see the introduction slide.
I have a problem with my presentation, so let me just check. Here?
Just to combine the...
Yeah, working with two screens is a mess sometimes. It's correct now?
No, it's still in the intro slide.
I don't know why.
Now we see. You can keep it like this. Yes.
Okay, so as I was telling, we are more than 15 years of a production, still currently active, of various optical elements like single lenses, doublets, and triplets that at the end are integrated into a machine built for the retina scan that's directly used from doctors and optometrists in their laboratory. So this is one of Tecnottica's most longer collaborations since the company started to work in the precision optic manufacturing at the beginning with this project. So it's one of the first and one of the still active one that we are very proud of it, since this machinery once has been established into the market as a continuous selling and application over the years. Some other examples are plus 5 years production of complete optical head, so devices where optics produced by the company were integrated with various mechanical and electronical components in order to create a complete system for medical machine visioning, so also for eye and retina inspection. And another one that's it's good to mention, it's production of mirrors. Another one that is currently active at this moment, directly assembly into a medical ophthalmic machine, where the visit for the patient for the eye examination and the vision check happens into a kind of virtual reality environment. So we have developed and produced these specific mirrors for these applications. And there are many other collaboration that I can talk about. But since our production is custom based, so we'll rely to our customers' need and their projects needs in order to build various optical elements for their application. So again, you will see probably better during the September visit, but in the ophthalmic sector, our components, for example, are chosen because of our flexibility, but for again, for our very precise capabilities and our historical joining of these applications. So again, this was just a short brief of the company itself. We'll have more time at the next event. So in the meanwhile, thank you for the sponsorship again and for the time that you have dedicated us today, this afternoon.
Thank you very much, Alberto.
Thank you.
Well, we are reaching the end of the meeting today. I hope you enjoyed all the presentations. It's a quite broad topic, but we try to cover some interesting new developments and also some topics related with IOL. That is definitely a hot topic in the different companies that are developing solutions for the eye. So thank you again to all speakers as well. So Yanni, Samuel, Sila, Francisco, and Altan, and of course Alberto as well. Thank you for your presentations, for showing our technology, and I hope you enjoyed the meeting. If you find something that is interesting for you, you didn't have the time to contact this afternoon, or do you prefer to have a question more in private mode, please do not hesitate to contact me for connecting you with the speaker or one of the attendees. Feel free to drop me an email and I will introduce you to discuss more in detail. And again, thank you, and I hope to see you soon if I were in next online meeting and also in our activities during Laser World of Photonics. I hope I will see you in Munich, most of you as well. And yeah, have a good rest of the day and see you soon. Thank you very much. Thank you. Good afternoon, everybody. Thank you, Samuel. Thank you very much for the time.
Bye
Thank
bye.
you, Altan. Thank you.